Hello, everyone. Thank you very much for joining our seminar today. My name is Maria Grigoratou, and I'm a biological oceanographer with expertise in zooplankton ecology. I'm a science officer at Mercator Ocean International and at the coordination center of G7 Future of Our Oceans and Seas initiative. I'm very excited to moderate our today's seminar and learn more about the BioGOSHIP from our two speakers, Sophie and Luke. I will ask all the participants, please write your questions and comments in the Q&A box and not in the chat box, and all the questions we'll address at the end. Before I give the floor to our speakers, I would like to briefly introduce them, starting with Sophie. Sophie Clayton is an assistant professor of oceanography at Old Dominion University. Sophie received her bachelor degree in ocean sciences from Bangor University in the UK, followed by a PhD in physical oceanography from the MIT WOCE Joint Program in the USA. Her research focuses on observing and understanding fine-scale variability in phytoplankton populations, their interactions with physical features, and how they ultimately influence large-scale ecological and biogeochemical patterns and processes. Her lab is developing methods to improve the automated identification of phytoplankton from high throughput imaging instruments, as well as studies analyzing large environmental and ecological data sets to better understand physical and biological interactions in regions such as the Mid-Atlantic Bight, the California Current, and the Cohaisio Extension. Luke Thompson is an associate research professor at the Northern Gulf Institute of Mississippi State and NOAA's Atlantic Oceanographic and Meteorological Laboratory in Miami. Luke received his bachelor degree from Stanford University and his PhD from MIT, both in biology. His current research focuses on marine systems from microbes to fish to mammals, using omics methods, especially DNA sequencing. His lab is developing methods to facilitate high throughput environmental DNA sampling processing and data analysis, and applying them to monitoring and conservation efforts in the Atlantic Ocean, the Gulf of Mexico, and the Great Lakes. As I said earlier on, please write all your questions and comments in the Q&A box and not in the chat box, and all the questions we'll address at the end. So, Sophie, the floor is yours.
Thank you, Maria. Okay, let me just get this shared. Can everybody see my slides okay?
Yes.
Wonderful. All right. So thank you so much for the invitation to present to this seminar. I think it's a really nice opportunity for us to speak to some people in different places. I feel like we've been spending a lot of time talking about BioGOSHIP within the US, and I'm excited to try and spread the word to a wider audience. And so the talk today is split between Luke and myself. I'm going to give you guys an overview of the BioGOSHIP program, and then Luke will pick up about halfway through with some follow-on about more of the technical details of what we've been working on. So the title of the talk is BioGOSHIP, Expanding Biological Ocean Observations for Plankton Ecosystem Science and Monitoring to the Global Scale. As I said, I'm going to start with an outline of the BioGOSHIP program, and I'm going to give you guys some science snippets. So most of this talk is aimed at a pretty high level, but we'll go into a little bit of detail in some of those science snippets, some previews of work that we're doing and work that has been done based on this program. And then Luke will jump in and talk about some activities we've been doing around method standardization, data management, and integration with partners outside of the BioGOSHIP program, or within integration, basically. So this is truly a team effort. Luke and I are here today to tell you about this program, but there are many of us involved, covering quite a wide range of observational tools and methods within biological oceanography. So I want to introduce the team, and this effort has really largely been spearheaded by Adam Martini at UC Irvine, Manuel Boss at University of Maine, and Heather Benway at the OCB office in Hui has done a lot to help get this program off the ground. And then Harry Alexander at Hui is one of the people working mostly with omics. Myself, Sophie, I'm based at ODU. I'm working on the plankton imaging side of things. Jason Graff at Oregon State University is leading up the biooptics piece. Nicole Poulton at Bigelow Marine Labs is leading up the flow cytometry, and then Luke is working with omics and eDNA, and you'll hear more about that later in this talk. And we've had some preliminary funding from NOAA, NASA, and NSF, and we're working hard to integrate within the global GOSHIP program, but I'll tell you a little bit more about that as we go along. So the focus here is on phytoplankton, and we know that phytoplankton are forming the base of the ocean food web, and they're underpinning the blue economy. And this is projected to be worth about $3 trillion by 2030. Phytoplankton not only are the base of the food chain, but they mediate biogeochemical cycles of climatically important elements such as carbon, nitrogen, phosphorus, oxygen, and so on. So we really want to understand what is driving the activity of phytoplankton. Their metabolic potential, their activity are really recognized as an important knowledge gap, partly through lack of observations. And so understanding how phytoplankton can be impacted by climate change will require a baseline understanding of the current state of the ecosystem. And I would argue, and I'm sure many people would agree, that we don't currently have that baseline understanding. We do, however, have a baseline understanding of the physical system. And so I want to give you guys a quick introduction to the GOSHIP program. I don't know if everybody's familiar with GOSHIP, but GOSHIP is the Global Ocean Ship-Based Hydrographic Information Program that has been running in one form or another for at least 3 decades. And so the map I'm showing you here is a map of all of the GOSHIP lines. So these are lines that ships are running on a decadal timescale to take repeat observations of the ocean. And GOSHIP is organized around different levels of data. And so some data are required and then others are desirable to have. And what I've put on the right-hand side of this slide is the list of the level 1 data. This is the data that has been required by the GOSHIP program for some time. And I don't expect everybody to read the entire list, but what I do want to point out is that there are no biological parameters on that list of required data. So we have a great view of the physical structure of the ocean from GOSHIP. We have some great chemical data, biogeochemical data to some extent, but really not much in the way of biology. So I will say, I don't want to get into trouble. Biological data have been collected. They are sometimes collected, but they are not required measurements. Okay. But hopefully what you can see here is this is a great platform. We have a platform of decadal surveys covering every single one of the ocean basins, and also all of these measurements that are being taken already. So physical measurements, chemical measurements are already part of the GOSHIP program. So BioGOSHIP aims to leverage the existing GOSHIP program. And so the vision of BioGOSHIP is to develop a deep understanding of the link between the physico-chemical environment that we can get from the GOSHIP data to the diversity of global ocean plankton and their biogeochemical roles. And that will give us some sense of how they will play into the changing ocean. So I gave you guys a view of GOSHIP. I showed you the map with the repeat lines and gave you the sense that there is a set of level 1 data, these required data sets that have that go through a certain amount of checking and QA/QC to make sure that everything is sort of that we're getting good data, that's intercomparable. And all of those measurements rely on mature technologies. And I think one of the things holding back biological observations for some time is either the lack of mature technologies or the fact that some of the measurements are quite labor intensive to do. And so I think now is a great time for BioGOSHIP to initiate, because we have many sensors and many tools that are actually quite mature. We have mature technologies that we can use to measure biological variables. And I'm giving you a quick overview here of some of the measurements that we have incorporated into the GOSHIP program. And they include sensors that are measuring data inline, so using water from the ship's underway system. They include things like flow cytometry that I'll talk about in a little bit, backscatter, chlorophyll fluorescence. We can also collect samples from the CTD and have sensors on the CTD. So again, chlorophyll fluorescence, backscatter. We've deployed a UVP. That's an underwater video plankton recorder. We have lab on land, so we can collect samples at sea and then take those samples back into the lab for genomic analyses, for HPLC analysis, for flow cytometry and eDNA. And then finally, many of these research ships have ADCPs on board or echo sounders, so we can use the acoustic data to try and get at some of the information on higher trophic levels. So we have a set of mature technologies that we can deploy as part of a repeat hydrographic program to target biology. So again, the vision is to integrate these core BioGOSHIP hydrographic chemical measurements with these BioGOSHIP measurements that will give us information on plankton biodiversity, on biological stocks, and sources and sinks of chemical and biological variables. So we now have the potential for a very detailed observational characterization of the ocean environment. And we see that this is going to be a kind of a back and forth between the core GOSHIP program and the BioGOSHIP program to collaborate on some of these questions. So we also have some science questions, and we again feel that this combination of the observations from the mature technologies over the global scale from BioGOSHIP tied back to the very mature GOSHIP program will allow us to answer some important science questions that are still to some extent, or to a greater or larger extent, open questions. And those relate back to patterns of plankton taxonomy and functional diversity, the impact of physical processes in driving biogeography, biodiversity, the role of plankton in global biogeochemical cycles. We're also very interested in trying to understand how carbon export fluxes vary and how they can be impacted by the surface ecosystem. And then finally, what are some of the global patterns in the plankton size spectrum? And these are all things that we can address with the measurement methods that we're deploying as part of BioGOSHIP. So we are in the process of running the BioGOSHIP pilot project. Again, this is funded through NOAA, NASA, and NSF through the NOC program. And this map just gives you a sense of where the GOSHIP sections are. So the light blue lines are all of the GOSHIP lines on this map. The red sections have been finished, and the orange sections show the sections that are part of the pilot program. And so we've just completed section P2, which crosses the Pacific from Japan to California. Early next year, we will be joining section A16N, which will go up from Brazil to Iceland. And we have several proposed sections in future that are not part of the pilot project, but we will be actively seeking funding to make sure we get onto those proposed sections that are shown in black on this map. So hopefully you can see that we can really get global coverage, and we also have the opportunity to go to places like the Indian Ocean, where there's really big gaps in the observations of ocean biology. Okay, so this is for those of you who are really interested in gear. So this is the setup that we had on the Ravel for the P2 cruise that happened from May to July of 2022. And so we have many instruments set up, all plugged into the underway system here. We have a hyperspectral flow cytometer, we have NACS, backscatter meters, we have a part of the bench that's set up for filtering. But I think the point I want to make here is that the capacity exists on these cruises that are already funded to do the physics and the chemistry. And we can very easily set up this BioGOSHIP sampling setup within a relatively small footprint, so long as we have access to the underway system, to the flow through water. We're good to make lots of very valuable measurements with this setup. So I'm going to show in a minute some preliminary data that was collected with this setup on cruise P2. But before I get to that, I'm going to also talk about some results from Adam Martini's previous work on some of the cruises that were done before the full-scale BioGOSHIP program started. So some science snippets. I wanted to give some examples to cover 3 of these main points that I think are worth talking about. So we're interested in metabolic potential of the plankton. So I'm going to show you some data from Adam's lab, looking at global patterns of nutrient limitation in Prochlorococcus. We're interested in biodiversity of the plankton. And so again, some results from Adam's lab, looking at patterns of global ocean bacterial diversity. And then finally, I'll show you some of those preliminary results from cruise P2, where we're trying to get at the activity of the plankton. And we have some preliminary data looking at the diel cell cycle of Prochlorococcus and Synechococcus. So this is some of Adam Martini's work. It was published in 2021. Ustik and Larkin, I think it was Larkin, either in Science or Nature. And this shows you a global map of nutrient limitation of Prochlorococcus in the surface ocean. And this, I think, is a great illustration of the power of collecting data from these global repeat cruises. There are some GOSHIP cruises in there in the Indian Ocean, but there are also some data collected through the AMT program and some other opportunistic cruises. But you can see really coherent patterns here in the limitation of Prochlorococcus in different regions, as well as patterns of co-limitation. And this was all done based on collecting samples at sea and then running metagenomic analyses of those samples back on land. So this is very doable in terms of getting the samples and then bringing them back for the analysis. And I think this is really exciting to me. I see this and I think of all of the ways that this work can be extended by tying back some of that environmental data and then thinking about how it could be brought into programs like the BGC program and thinking about remote sensing and validation of different products. This is super exciting to have this large-scale view of nutrient limitation from samples taken within the ocean rather than as an inference from other data types. So another example here from Adam Martini's lab. This is a map of bacterial ocean biodiversity. So again, you can see the map where all those samples came from. It's the same as in the previous slide. And then this Shannon index map on the left shows these patterns of diversity. So a low number on the Shannon index is low biodiversity, a higher number in red is high biodiversity. And there are some pretty consistent patterns of low diversity in the low latitudes, increasing as you go into the higher latitudes. And what Adam found, and this is work that he presented at the Ocean Sciences meeting earlier this year, was actually the best predictor of bacterial ocean biodiversity is not anything like eddy kinetic energy or dispersal, but is actually the abundance and the presence of Prochlorococcus and the extent of nutrient stress. And so this sort of points out that there is one organism that's really driving these patterns of biodiversity for bacteria in the ocean. Again, I find this, thinking of where this could go is very exciting. All right, and then the last science snippet. This is some very preliminary data that was collected during this P2 cruise. You can see the map there on the left-hand side. This crossed the Pacific from one end to the other. And we ran a flow cytometer continuously throughout this cruise. So Nicole Poulton set up a SciTech hyperspectral flow cytometer, and we've started looking at that data and pulling out the abundances of Prochlorococcus and Synechococcus. We can get at the cell size, the equivalent spherical diameter of those cells, and then we can look at the distribution within any given time point. And so what we've done here is to pull together data from a couple of days for Prochlorococcus and Synechococcus. And hopefully in these graphs, you can see as the day progresses, this diel cycle in the cell size distribution of these two important cyanobacterial organisms. And the goal is to use this data to derive rates, division rates, rates of carbon uptake, and rates of carbon loss from this continuous flow cytometry data. So I'd say watch this space for this one. We're hoping to have some more robust results for the entire basin sometime next year. So I've given you an overview of the program. I've shown you guys some snippets of the science that we've done and we're doing, and I'm going to hand over to Luke now, who's going to talk a little bit more about some of the nuts and bolts of making this program work in a sensible way.
Thank you, Sophie. Yeah, that was a great introduction and description of some of the preliminary results of the program. And so I want to talk a little bit about the methods that we're using and how we're standardizing and optimizing them, as well as how we manage data from this program. So it's an incredible amount of data from all these different sources and these methods. And so how we manage that data is really critical. And then finally, and this is related to all of the things that we're talking about, is partnerships and how we're working with partners and talking to partners and collaborators about all of these things, about how we measure the ocean, how we manage our data, et cetera, and how we're making the most of that. So to start, I'll talk about an area that's close to my expertise, which is eDNA methods. And when I say eDNA, I'm talking about environmental DNA or any source of DNA that's in the environment. So this is could be fish or whales or plankton or anything down to bacteria, and also includes free DNA. And I'll talk a little bit about what kinds of sources of DNA there might be in the ocean. But what we do with the genomics work and the eDNA work in this BioGOSHIP project, and this is similar to any other environmental survey, is we collect the sample, we filter that via either vacuum or peristaltic pump. We then, back in the lab, we will extract the DNA from the filter, and then there's some form of library preparation or PCR to either amplify or prepare the DNA for sequencing. And then there's the DNA sequencing, or in some cases, a quantitative PCR analysis. And so we've got a lot of these methods pretty well settled. So sample collection is either done using a CTD rosette shown on the bottom left, or we also use the flow-through system. On GOSHIP, we're using the flow-through system that Sophie showed earlier. We're using that a lot because there's such a high demand for water on these cruises that we can't always get deep water. But we're hoping, and on previous cruises, we've been able to get around one cast per day, one water column profile per day. And so we're hoping that continues. So there's two sources of water, either from the CTD or from the flow-through system. The filtration is done using a peristaltic pump, and that's shown in the next little pane there. And then that filter, we're using a StereoVex filter, which is this plastic cartridge filter that's shown in the center bottom. And we're working on different ways to extract that, and I'll talk about that in the next slide. And then there's different ways to analyze the data. So the main methods we're using, we're not doing so much quantitative PCR, although that's an option. We're doing more metabarcoding, where we sequence a single gene from all of the organisms, say all of the bacteria or all of the fish or all of the plankton, and we sequence and analyze that. We're also doing shotgun metagenomics, which is total DNA sequencing, and the data from Adam Martini's lab that Sophie presented a few slides ago, that's predominantly from shotgun metagenomic data. Next slide. And so I'll talk a little bit about the DNA extraction optimization that we're doing and standardization. And so in seawater, there's different sources of DNA. Could be from cellular matter, tissues and waste, or single intact cells. Could be also decaying cells, dying cells, they still have DNA. Or there could be free DNA that's been released from different sources floating in the water. So wherever the source, we can, if it will be filtered onto a 0.2 micron filter, we'll be able to collect that and extract that. So I'll talk about the extraction in the next slide. And so there's three methods that we're currently comparing. This gives you just an idea of the kind of detail that we're going into to make the most of the samples that we have. So there's at Martini's lab, which is the current method, they're doing a extraction inside the filter with a few different enzymes and then a precipitation step. But there's a couple other methods we're trying. So Harriet Alexander's lab is doing a method where they remove the filter from the cartridge and extract with a kit from a company called Zymo. And then my lab is doing a method where, like the Martini protocol is inside the cartridge, but we add beads and a lysis buffer and we agitate that. And then we use this, this robot shown on the lower right called the Kingfisher that allows us to do high throughput extraction using a magnetic bead kit. And so we're comparing these at the moment and going to decide on the most efficient and reproducible way to move forward. Next slide. So next, I want to talk a little bit about data integration and management. So as Sophie showed, there's just so many kinds of data and methods that we're using. And so we really want to make the most of this data. And so if you just look at a single type of data or want each type of data in isolation, they're going to have limited coverage, and it's only going to show you a little, narrow view, maybe a very detailed view, but perhaps a very narrow view of the ocean and its biology. So it allows limited inferences. But if you're able to combine different data types, that allows a more holistic and kind of synergistic understanding. So these data sets can play off each other, and you can really get the most of each one in combination. So how do we do that? So I've kind of summarized the challenges of data integration as data must be better curated. And so what do we mean by that? So we've got diverse data types. And so one of the ways that we can integrate these is to process them into a shared format, basically like a tabular format. If you can get the data into a tabular format where you have samples as rows and your columns are different types of observations about those samples, then you might be able to apply some of the more general data science tools to do that. But that requires some decisions about how you take this very rich data, whether it's image data or omics data, and then condense that into a single number or a set of numbers. Another challenge is incongruent sampling or gaps. So either maybe a particular instrument wasn't functioning properly on a given day, and so you have a gap in the data, or just perhaps because of the realities of working on a cruise ship, on a research vessel, you might not be able to get the same data sets from the same exact samples. And so you've got to deal with these sort of incongruencies. And there's things we can do like interpolating data and other things, but that's definitely a challenge with data integration. Another one is standardization. So we want the data to be standardized. If it's in a all the given data from a single type of data, if it's in the same format, it's much easier to analyze alongside other data of that same type. And then it's also therefore easier for different data types to sort of talk to each other if they're all in, each one is in a standard format, and they're ideally in a congruent format. So for example, if one data set has latitude in degrees, minutes, seconds, and another data set has latitude in just decimal degrees, then there's some work that has to be done to get those to match up. And maybe there's rounding errors, and you can imagine this is the kind of stuff we deal with data. So the more we can standardize things up front, the easier our life's going to be when we really want to get the most out of the data. But there are challenges to that as well, because there are sometimes incomplete standards, or there's multiple standards, and we have to decide. And finally, sharing. So we want to make the data accessible. We want to put it out to the public so that not just our group, but that the world can use these data. And so there are repositories for that. So NOAA has the National Centers for Environmental Information, or NCEI. And then we're predominantly a US-based group, so I'll talk more about the US agencies, although there are European and international versions of these. There's also the NCBI, which is the National Centers for Biotechnology Information. That's through the NIH. And then there's the Ocean Biogeographic Information System, or OBIS, which is a biodiversity facility. And so these are great resources, but it's still incumbent on the generator of the data to put those data in there in a timely fashion and make sure that there are links to the raw data and to the metadata or environmental data. Next slide. And so there are a few things that we're doing. I mean, some of the solutions kind of come naturally from those challenges, but there's a few other things that we're doing. So making sure that the requirements are well defined. So you have mature standards that are agreed upon by user communities, and we're involved with several different international and national groups that are working on these data standards. Training resources, so setting up workshops, talking to each other, and also developing online websites and wikis. These are really useful to educate people. And also, I have here, preaching the FAIR data principles. So the FAIR is an acronym that stands for findable, accessible, interoperable, and reusable. So we want people to really embrace this ethos of making the data easy to find, easy to use, interoperable with other data sets, and then reusable. And then another thing that we're exploring is positive incentives. So things like giving a data set a rating, say, if it's really useful, if it's really well put together, you can get a 5-star rating. This hasn't really been rolled out yet, but this is an idea that's been tossed around. Also, having citable data DOIs or digital object identifiers. So these are ways to give people credit for their data sets, even if it's not in a peer-reviewed publication of a manuscript, say. Next slide. And so to develop some of these resources, we've been regularly meeting, and we had a data workshop in October of last year with BioGOSHIP PIs and other members of the group, people from the different data repositories and our federal sponsors. And so from this workshop, we started a dialogue and information exchange for this pilot project that Sophie talked about. We shared different information about the different observations that we're using and what are the best practices and what are our needs. We mapped out some of the data flows and the different parameters that we're measuring. We talked about repositories. I mentioned that in the previous slide about the different repositories that are out there, and discussion of standards as well, and identified topics for a deeper discussion. Next slide. And so there's some working groups that have been established from this workshop. We have a data requirements workshop, a data requirements working group to ensure that we're following the GOSHIP reporting standards and following best practices. A data repository working group, obviously about different repositories that we can use. And then a global marine plankton biodiversity working group, which is a little more advanced, I guess, or more requires some deeper investigation to figure out what's the best way to integrate all these different data sets to get a more holistic understanding of plankton biodiversity. So some of like Sophie showed a Shannon diversity in one of Adam's slides. So that's a summary metric, but there's a lot of other kind of things. And so this is something that we're actively working on. Next slide. And so as an example of that, from the omics data, this is just shown by a DNA molecule, we can match that data to a database and we get relative abundance of different groups. We can combine that with the flow cytometry data and get a sense of how these two different data sets co-vary with each other, and how you might be able to infer, say, one kind of data from the other, or where there might be incongruencies or missing pieces that might actually be interesting, might actually tell you something that we were missing before. Next slide. And then finally, there's a lot of partners involved with this. So we have a really great relationship with NOAA. The Global Ocean Monitoring and Observing Program has provided funding and really a vision for this program, and we're incredibly grateful for their support. Also, the Japan Agency for Marine Earth Science and Technology is a partner, and they're involved with various aspects of the international GOSHIP program. We're also coordinating with other networks. So several of us are involved with the Marine Biodiversity Observation Network, or MBON, and just like BioGOSHIP, MBON is developing observing systems and dealing with data integration and data management and all these very important and related issues. So we're really trying to leverage those relationships. There's also the Ocean Best Practices System. This is a international group that's helping to develop various aspects of ocean research, and with regard to BioGOSHIP, protocol management, data management, and standardization are a big part of that. And then this is currently the UN Ocean Decade for this current decade, and there's a related program called Marine Life 2030, and I'll talk about those in the next few slides. So the UN Ocean Decade has a few different challenges that are relevant to BioGOSHIP. So one is to protect and restore ecosystems and biodiversity. And so obviously we can't protect and restore that diversity if we don't know what's there. And so just having a basic understanding of that is where BioGOSHIP can really help a lot. Expand the global ocean observing system. So clearly that's an important link to BioGOSHIP. And then developing skills, knowledge, and technology for everyone. And so we're here today talking about the program, and we're supporting graduate students and postdocs and sharing knowledge as much as we can to really tackle this incredibly important and big problem. And so there's a particular program that the UN Ocean Decade has endorsed called Marine Life 2030. And so a GOSHIP plankton observation program like BioGOSHIP fits really nicely with that, as I'll talk about in the next few slides. So this is just a title slide for Marine Life 2030, building global knowledge of marine life for local action. And this is part of, I said the UN Ocean Decade, but also MBON is a partner, and the Global Ocean Observing System, or GOOS, is another partner. Next slide. And so, and this is kind of bringing it back full circle to what Sophie started with. There really is not at present a systematic effort coordinated internationally to observe life in the sea. And so a goal of Marine Life 2030, and just as it is a goal of BioGOSHIP, is to enable efforts to characterize how marine biodiversity is changing, how it affects us, and how to guide sustainable development. Next slide. And so, again, before we had programs like BioGOSHIP and some of the others like Tara Oceans and the Global Ocean Sampling Expedition, there hasn't been, and really to this day, there is not a sustained global monitoring of biodiversity beyond chlorophyll. So this is a really nice form of data. This is satellite-derived chlorophyll concentrations, either in the ocean, is what we're concerned with, of course, is chlorophyll A concentration from NASA satellite data. So this is a wonderful data set, but it's limited in what you can learn, right? It doesn't have all of the depth information. It doesn't have all of the biodiversity information. So how do we move beyond chlorophyll? And that's really what BioGOSHIP is about. So next slide. And similarly, just to illustrate this point, the Ocean Biodiversity Information System, or OBIS, that I mentioned previously, this is a repository for biodiversity data. And if we go in and look at OBIS, there is a lot of data in there already, but it's concentrated toward coastal regions, the North Atlantic, and a few other regions. It's concentrated toward more the surface, and there's a drop in some of the higher latitudes, as well as in recent years there haven't been as many submissions. So there's a lot of gaps, of course, in this data set, and so there's a lot of opportunity to add data and to really fill out the map, as Sophie was showing in some of her slides. Next slide. And so just to wrap up, and we'd love to have questions and discussion as there's time. Over the next 5 to 10 years with the BioGOSHIP program, we envision a global high-resolution omics, optical imaging, and elemental stock measurement system that will enable observation of ocean biology on a global scale. We want BioGOSHIP to be a testing bed for new biological and biogeochemical sensors, for example, an underway ESP, which is an environmental sample processor. And we want to make BioGOSHIP data freely available to the community and provide a rich global set of biological data for data mining, model, and satellite validation. An example of this is the PACE program, which is a fisheries-related effort. And so with that, appreciate your attention. And yeah, we'll just say thank you, and we'll put our slide back up there with our faces and the all of the sponsors that have supported this work.
Great. Thank you very much, both Sophie and Luke, for this very interesting presentation. And we already have some questions in the Q&A chat. I will ask the attendees to also add some more or comments if they have at the Q&A chat. So I will start with the first questions from Elva Escobar. So we have one question: are the data collected in exclusive economic zones shared with local scientists?
I guess I can try and field that one, and then Luke, you can jump in if I get stuck. So in order to be able to collect data in EEZs, we have to request permission, particularly for biological data. We won't necessarily always get it, but if we do, any data collected on BioGOSHIP is shared. So there's no difference between the data that we collect in the middle of the Pacific and the data that we collect off the coast of Japan, for instance. So it's all shared in the same way. So I guess the answer is yes. If we do collect any data in EEZs, it will be shared and made available to everybody in exactly the same way that any data collected outside of the EEZs is.
Okay, then we have the second question. Does the BICSI program help build capacities for developing nations?
Luke, do you want to field that one?
Yeah. So I guess explicitly, the primary support is to get the observations done, and that's the primary focus. But there's a lot of we're very open and kind of sharing focused group. All of the resources that we're developing, in terms of the protocols and the data standards and the data analysis methods are open source. So my group has a big GitHub organization and with a lot of repositories with how we analyze our data, and the data management materials are available as a wiki on GitHub. And so we really try to make these as freely available and as clear and usable as possible. And I think that definitely does benefit developing countries where it would be harder to pay for licenses or anything like that. And I think as we continue and we develop training materials and workshops, I know there's a few people from Brazil here. I was just in Brazil last week on an MBON project. This is the MBON pole-to-pole project, which is a sort of a global effort to bring together different researchers from different groups. And we were meeting with researchers in Brazil and sharing knowledge, discussing some of the issues with data standardization and data sharing, and a lot of the same issues that we're doing with BioGOSHIP. So there's a lot of conversations happening, and actually, I mean, this Networking Friday is a great chance to share what we're working on, and if there's any way we can do better about making things available and sharing that, we would love to improve.
Thank you. Then we have the last question from Elva Escobar. What is the cost for the average library preparation and DNA sequencing?
Yes, so it varies depending on how much you multiplex. Generally, I would say it's about $50 a sample, $50 to $100 a sample. It depends on a few factors: how many different types of sequencing you do, how many samples you're able to sequence together in the same sequencing run, and if there are anything that doesn't work, maybe you have to go back, and that can add to the cost. But yeah, I'd say on average about $50 a sample. So it definitely adds up. But with some of the methods that we're developing and combining more samples, that some of these new sequencing platforms can sequence a lot more in a single run. So I think that cost is going to come down.
Great, thank you. Then I have two questions from Frank Muller-Karger, which I would like to combine them, and then I can give you the floor to answer them. So first, Frank asks if it's, maybe it's already happening, but if it's possible to coordinate on data mobilization workshops with OBIS EMBON, that they have a workshop next spring, and with BioGOSHIP. And then the other question is maybe if there's an interest in seeking endorsement from the UN Ocean Decade for BioGOSHIP as a program affiliate with the Marine Life 2030.
I would venture to say that the answer to both of those questions is yes. I think that if we can coordinate with the OBIS EMBON data mobilization workshop, that would be fantastic. And I guess that's just a case of us getting in touch. So thank you, Frank, for bringing that to our attention. I'm guessing Luke probably already knew about it, but I wasn't aware. So that sounds fantastic. And I think, I think seeking endorsement from the UN Ocean Decade would also be a great next step for us. We've been a little bit focused on getting everything up and running for the cruise in the Pacific earlier this year, and now we're gearing up for our next cruise in the Atlantic. So we can be a little bit scattered, but certainly any involvement in those programs I think would be fantastic, and we'll aim to do that.
Great, thank you. Luke, do you have anything to add, or shall I move to the next question?
Yeah, no, I think definitely would love to make more partnerships, and Frank, perhaps at one of the upcoming meetings we can have a little presentation from the BioGOSHIP team and talk about what makes the most sense.
Great, thank you. Then we have a question from an attendee, which it's anonymous. Is expanding plankton observation by including continuous plankton recorder service feasible for the BioGOSHIP or the GOSHIP cruises?
You're going to have to forgive my ignorance, because I can't remember exactly how the CPR is deployed. And so I would venture to say that if it is something that can be run underway, sort of fed by the ship's flow-through system, then I don't see a good reason why it wouldn't be possible, but we might be limited in terms of flow rate, that kind of thing. If it's something that needs to be deployed from the ship, then I would say that the answer is most likely no, because the GOSHIP operations are very tightly coordinated. These cruises are doing dozens and dozens and dozens of CTD casts, and it's the timing is very sort of coordinated. So if it's towed, I see that, yeah, you're saying it's towed. Probably, I doubt we would get the GOSHIP folks to agree to us running a towed instrument. We could ask, but I'd be less optimistic about getting the go-ahead on that.
Great, thank you. So now we're moving to some questions from Maria Pachiadaki, which is around data sharing and data protocols. So first, Maria would like to say thank you for great presentations. And then the community has been struggling for a year to optimize DNA extraction methods. So we have two questions around that. First is, how are you planning to evaluate the performance of each protocol? And then if you're using complex morph communities to grow truth. And then there's another question that I would like to add here again from Maria Pachiadaki, because I think it fits. Luke, you mentioned in your presentation that there is a time fashion for the data. Can you share more details? What is the time fashion for the submission of the BioGOSHIP data?
Yeah, good questions. So for the second one, a general rule of thumb is within a year of generating the data, it should be made public. Another rule that people generally follow is once the data are published, they should be once there's a manuscript about the data. But since we don't know when that will happen, I think within a year seems to be a reasonable amount of time. There's some people have may have concerns about sharing their data before they've had a chance to publish on it, but I think in practice that's not doesn't usually become a problem, and it's on the other hand, it's really useful to get the data out there, and it forces you to make sure your metadata is in a good shape as well. So I would say if possible within a year of data generation would be ideal. And then with the intercalibration exercises with eDNA or with the DNA extraction methods. So what we've been doing is trying to isolate the DNA extraction method from everything else. So we have replicates from the PO2 crews that were given to the 3 different labs to then extract and then send the DNA back to the Martini lab at the Irvine. And so there's less, it's not that each lab is doing the sequencing themselves. And so the plan is to do the shotgun metagenomic sequencing in the Martini lab, and then to compare the communities that come out of that. So different statistical methods to do that. And I think there was a question about mock communities or standards. So that's definitely something that we use in different situations. In this case, we have not used those kind of mock communities. It's difficult with marine samples to get the ones that are commercially available are more like human gut type of microbes. They don't represent really any marine organisms, whether bacteria or plankton. So I do think there's a need for some kind of standards that we can all share. And there's different ideas out there about what's the best way to do that, whether it would be you make giant vats of cultures and mix them together in defined proportions and everyone uses the same one. But what happens when you run out? Another idea would be to use like plasmids or some kind of like DNA that you can synthesize in large amounts and mix those together and share that with groups. This is something that we've thought about and have some ideas, but haven't really deployed it in any real way yet. So yeah, that would be a great improvement. But I think just having these 3 different groups analyzing the same samples, replicates of the same samples, and then sequencing them using a single platform is going to be helpful.
I wanted to circle back around to the data availability, because I think what Luke talked about is correct. So GOSHIP has particular timelines for different levels of data, and it ranges, I think, from 6 months to 2 years, depending on whether it's level 1 required through to level 3 desirable. I guess the one point I want to make, and this is going back to talking about sharing and best practices, the best practices for imaging data, for example, are still actually actively being worked out. There was a very recent effort sponsored by the Ocean Carbon and Biogeochemistry program at WHOI that put together a paper that came out only this year on best practices for sharing images from imaging instruments like the IFCB, for example. And there are still discussions needed with the repositories and the data managers, because again, you know, and I'm more versed in the imaging side of things, but there are still questions about actually how do we share these sometimes huge data sets, particularly images from IFCB and UVP, and then also how do we coordinate with the existing flow cytometry repositories that are not always environmental, sort of set up for environmental data sets. So I think I would hesitate to give an exact number right now, but our goal is to share the data as soon as we possibly can. But there's also work that needs to be done in terms of actually developing robust pipelines and best practices from the instruments to the repositories. And that's work that we're actively involved in. And I personally am quite excited about getting done.
Great. Thank you very much. So officially we have 2 more minutes for a session, but I cannot see any other question or comment in the Q&A box. Please feel free to add something if you want. I have one last question. I think this is an amazing effort. And I want to ask you, how can you include members from the international community to join on the design or in the sampling efforts, or in the decisions on data standardization and data sharing? Is that open to the community? And what would you like to get from the community? What do you need?
I could say, I think with sampling, you know, as Sophie showed, that there's a lot of cruises. We have the pilot, and then, you know, if things go well and we find additional funding, we can do more cruises. So there's going to be opportunities for people with some cruise experience to come out and help us on these cruises for sure. You know, I think we're still figuring out, you know, how things are going to work with all these different people involved already. So we'd love to, you know, involve as many people as are interested, but there's a coordination challenge. So I think, but I think if anybody has, you know, interest or a passion for an idea for this, you know, we would of course love to hear about it, and you know, there might be opportunities for pursuing that. But I don't know if Sophie, if you have any thoughts about that international integration and partnership question.
No, I don't. Yeah, I think you're spot on. We're to some extent in the early stages of this program, and it's super exciting. And the vision really is to be inclusive and international. I think at this point, what we're trying to do is to initiate coordinations with the international GOSHIP program, and that's still very much in the early stages. So we started coordinating with JAMS Tech with folks in Japan. But I think, you know, if people are interested, please feel free to contact any of us. And we would love to explore opportunities as they come up and have these conversations. So I don't think that there's we can't give a formal answer about how these things might happen, but we're very, very open to, you know, our aim is a global program, and that's going to require international coordination. So yes, please, you know, talk to us. We'd love to have these conversations.
Great. Thank you very much. I cannot see any other comment or question at the Q&A box. So I guess now we're on time to finish the seminar. Sophie and Luke, thank you very much for the very exciting presentations and the discussion. And I would also like to thank all the attendees, either from the people who joined from the Zoom's link or on YouTube. Thank you very much for being here today with us. And yes, I hope we will see each other around in another science meeting in the future. Thank you very much.
Thank you, Maria. Thanks for having us.
Yeah, thanks for having us. Have a great day, everyone.